Hello, everybody, and welcome back to BioSC 140. This is for the discussion of Lab 3, plasma concentrations. So let's look at our goal of this experiment. Our goal is to find out the concentration of glucose, the concentration of cholesterol, and the concentration of protein in our samples of plasma. What's plasma? Plasma is the liquid part of your blood. Your blood has the liquid part, plasma, then it has the white blood cells, the red blood cells. Plasma is the liquid part, so not the red blood cells, not the white blood cells. What is the concentration of these three things within your sample's blood? All right, moving on. So let's do some background information before we really get into exactly what's going on with this experiment. If you haven't watched the last video, you should definitely watch the data and experiment video for this. I'm going to assume you've seen it and you know it all. So plasma concentration composition is highly regulated. So there's a lot of different chemical reactions that how many chemical reactions need to take place inside your body? The answer is a lot. A lot of different chemical reactions need to take place inside your body. And in order for all of those chemical reactions to take place, there needs to be a very specific environment, a very specific setting in your body in order for all those chemical reactions to take place. Your body will regulate various variables in order to maintain an internal environment that allows all these chemical reactions, all these chemical processes to move forward. Glucose, cholesterol, and protein levels within your blood are three variables that your body regulates in order to maintain this ideal internal environment. Plasma levels of glucose, lipids, and proteins are held within narrow limits. Look back at Lab 2 if you're unsure what negative feedback is, or dynamic equilibrium. All three of these variables exhibit a dynamic equilibrium around a set point via bidirectional negative feedback. If any of those terms I just said or you're unsure of, revert back to Lab 2, consult Lab 2. Abnormal concentrations of these variables leads to a disease state. It is a disease state. It's pathological. Physiology is the study of how the human body works when everything's going right. Pathology is the study of when things go wrong. And you need to know how things go right before you can really get into how things go wrong. But we'll be talking about some pathology in this class because it can help illustrate and illuminate some ideas within physiology. So abnormal concentrations, abnormal internal environment is a disease state. I want you to know the normal ranges for these three variables. Pay attention to the units. I have thrown in questions before where I've changed the units and it usually gets a few students. So definitely know normal range, definitely know pay attention to the units. That's fair game for the test. When glucose is elevated, when it's higher than normal, it's called hyperglycemia. So hyper above, glycemia referring to glucose. A disease state, a disease process that typically leads to this is diabetes mellitus. Diabetics have a hard time lowering their blood sugars without medical intervention. My brother's a diabetic, and he is an insulin-dependent diabetic. When glucose levels are too low, my brother gets a little grumpy, but it's called hypoglycemia. Hypoglycemia. When your blood glucose levels are too low, it's called hypoglycemia. This can actually lead to syncope, which is fainting, or dizziness, or my brother being crankier than normal. Cholesterol. Know the range. It's called hypercholesterolemia. So hyper, above, cholesterol. Emia means presence in blood. presence in blood. So high cholesterol presence in blood. Hypocholesterolemia, too little cholesterol presence in blood, not too common. With protein, we've got hyperproteinemia, which can lead to some fatigue, and hypoproteinemia. Hypoproteinemia can lead to something called edema, which is basically you have too much fluid outside of your cells and outside of your circulatory system in a place called your interstitial fluid. If you've seen old people, like geriatric people, and they have really fat ankles, edema. So the ability to accurately measure levels of glucose, cholesterol, and protein is important for clinical analysis. The levels in healthcare, doctors measure and healthcare providers measure these variables all the time. Every day they're doing it. It's very important with modern medicine. So now that we know kind of where we're going, what we're looking at, let's look at what we're doing in this experiment to calculate our unknown variables. So we collected our plasma. That was step one. We used the centrifuge. We used the capillary tubes. We then put that sample, that plasma, into a tube with something called a color reagent. Now, there's a different color reagent for protein, a different one for cholesterol, and a different one for glucose. What the color reagent's gonna do is for glucose, it's gonna bind to glucose, and then that molecule, after the binding has occurred, will absorb light at a specific light frequency. So if the color reagent does not bind to glucose, it does not absorb light at this specific frequency. If the color reagent does bind to glucose, it's going to absorb light at a specific frequency. So the more glucose there is in the sample, the more binding to the color reagent there is, the more absorption of light there's going to be. So the spectrophotometer, what it does is it shoots light through our sample and it measures the amount of light that goes through the sample. So our blank should have no color reagent binding. We're going to get zero absorption on the other side. Then the more plasma there is, or the more glucose there is for the glucose sample, the more glucose there is, the more light the sample is going to absorb, the higher the absorbance reading from the spectrophotometer. The ratio for the amount of light that goes through, Beer's law, we can compare the absorption rate of a known substance with the absorption rate of an unknown substance, and we can calculate the unknown concentration. So Beer's law. Beer's law can determine the unknown concentration from a known standard. So you should be comfortable determining concentration using Beer's law, given absorbance readings with standard values, and then determine the consequence based on their normal values. So you should be comfortable using Beer's law. During our experiment, we were able to know the concentration of the standard. It was given to us. It's over here on the right. We should be able to know the concentration, we should be able to know the absorbance of the unknown. We use the spectrophotometer to get the absorbance of the unknown. We should be able to figure out the absorbance of the standard because we put the standard in the spectrophotometer. So we have 3 of these 4 variables. The only variable we don't have is concentration of our unknown. Well, we can do some math and we can figure that out. You should be comfortable using Beer's law. Here's a typical question I would ask for this lab. You're in lab and you're using the spectrophotometer to calculate the concentration of your patient's glucose, patient's plasma glucose. To do it, you use a standard that is 100 milligrams per deciliter, whose absorbance was 0.25. The absorbance of your patient sample was 0.3. What is the concentration of your patient's of your patient's sample, your patient's plasma glucose. 1 point for getting the math correct, using the Beer's law equation correct. 1 point for getting the concentration correct of your patient's blood glucose levels. Then I'll follow up in the question by saying, is this within the normal range? So 1 point then, 1 additional point for knowing if this was within the normal range, the value right here. Then I'll follow up that again by saying if this is outside the normal range, what is the vocabulary term associated with it? 1 additional point. Hyperglycemia, hypoglycemia. Right there, 3-point question. You should be comfortable with that type of question. All right, so here's some sample data that we can use. We have our standard, perfect. Oh, blank. So I want to talk about the blank a little bit because the blank is important. So when we set up the spectrophotometer, there will be a very tiny amount of light absorption because of the cuvette and just the water within the color reagent itself. We're going to put the blank into the spectrophotometer, and we're going to set that amount of absorbance as 0. So the blank is always going to have an absorbance of 0. It's always going to have a concentration of 0, right at 0. You can practice with sample data. Right here is sample data that you can use. Practice with it, calculating the unknown concentrations for your, this would be the student sample, this would be the normal serum, and this would be the abnormal serum. Here's some samples, perfect. Here's more sample data from the book. More sample data that you can use for, that you can practice with for glucose, for cholesterol, and for protein. Please practice before the quiz. Quizzes are timed. Quizzes are timed. All right, so let's talk about the standard curve a little bit. Let's talk about the standard curve a little bit. It might be hard in a time test to ask you to make a standard curve, but make sure you know how to read a standard curve and understand kind of the process behind it. Maybe I'll show you a photo and ask you to type something up describing it. I'm not exactly sure yet, but standard curve, you're gonna make a plot. We got the y-axis with absorbance values. We've got the x-axis with protein. We're doing protein for this concentration. These dots you see, those are the plotted data points for the known concentration for the standards. So we have multiple standards. We know that at, what is this, probably 2.5 or 2 concentration, it's 0.1. So we've got our known samples right here. Occasionally in lab, we'll have, we have 5 data points for protein. We'll have 4 data points that form a nice, straight, perfect line, and 1 data point that's like way up here, way out here, that's an outlier. There was probably some experimental error at some point. If there's an outlier, if you have 4 really good ones and an outlier, just ignore it. It happens sometimes in lab. You want to use a best fit line. You can see that the data points here are not perfectly straight, but they're pretty straight. When you form the trend line, you want it to go right through your data points. Plot only for standards. Do not plot for unknowns. Plot only for standards. We're going to use the standard line to calculate the unknowns. Let's say our unknown absorbance value is 4. We're going to go to 4, follow it straight across till we hit the line right about here, then straight down. At 0.4, I'd say that's about what, 10, 11, or 12 grams per deciliter. And that's how you figure it out. If it's 0.5 for the unknown absorbance, you follow the line straight over till you hit the line. Then you go straight down, looks like maybe 15 grams per deciliter. It's a good way. You should be comfortable making standard curves and using them to determine unknown concentrations. Note that you need 2 or more points to make a standard curve. So really protein, because you have 5 standards, is what you need, is the only one you can really do the standard curve for. All right, let's kind of review what I want you to know for this. For the quiz, I want you to be able to tell me the story of how we got the data. So from our last video, our data experiment video, tell the story of what we did. How did we get the data? Know what a spectrophotometer is. It's a machine that shoots light through a sample at a specific wavelength and measures the amount of light that gets absorbed by the sample. Know the normal ranges, know the vocab terms, definitely know the vocab words. It's really easy to write questions about that. Understand the importance of, of being able to know specific levels, like amounts of glucose and lipids and proteins within a person. They're regulated within a narrow limit. That's what I want you to know. There's a set point for these variables. There's an ideal value. And if you deviate too far away from that ideal value, you have a disease state. Understand, anytime you see an underlined word or a bolded word, you should know about it. So understand what the blank is. Why did we use a blank? Understand what a standard is. It's a sample with a known concentration that we can compare samples with unknown concentration to in order to figure out the concentration of that unknown. Definitely be comfortable with Beer's Law. You're gonna see Beer's Law either on a checkup or on a quiz. It's gonna be there. So be comfortable with it. Be comfortable with the type of question that I described earlier. All right, if you have any questions, be comfortable with the standard curve. If you have any questions, send me an email. I'm here to help.